basically if there is no catalyst, the reaction will take place. Maybe it will take some few years or few thousands of years, correct? Like this same way, biological components were synthesized on Earth. Ammonia, water. So it took years. Now we can synthesize water in the lab within a fraction of a second. But it took years naturally to form one molecule of water. Can we wait for those many years to grow up? We cannot wait. So biological system is faster than the natural system or slower? Huh? Faster, slower? Faster. So enzyme should be more efficient in increasing the rate of reaction than catalyst or not? Correct? So enzyme has to be more efficient than any catalyst. Any catalyst. Any catalyst you name, enzymes are at least 1000 times more faster than any catalyst. At least, if not more. Correct? On average, it is 10 to the power 6 times faster than any catalyst. 10 to the power 6 times. Now, these fast reaction because what? Any reaction. Because what? Proximity. Unless the two molecules are very nearby, they cannot, because molecules do not have emotions like human being. Like over the phone, if we hear someone has died, tears comes out. So telephonic reaction. It need not to be very close physically. So since molecules don't have emotions, so it has to be very close, nearby. Mostly the enzymes are protein in nature, mostly. Is there any other enzymes which are protein, no, which is non-protein in nature? Yes. The initial enzymes were all non-protein in nature, initial enzymes. Why? The initial molecule was nucleic acid. So nucleic acid was synthesized by nucleic acid. Then protein started synthesizing the components of nucleic acid. Right? So initial enzymes were non-protein. Later on, nature decided to fix one biomolecule for all enzymatic action, that is protein. But before that, whatever discovered were discovered by nature or discovered, remain as such. Right? So nucleic acids are called, nucleic acids are there which are non-protein but still they have enzymatic action. Say for example, the very important peptidyl transferase activity. That means in the translation process, the enzyme which is responsible for synthesis of earliest peptide bond. It is hen before or egg. Peptide bond before or polypeptide which synthesizes peptide bond. Which one? You should have peptide bond first or polypeptide to synthesize peptide bond first. Which one? Peptide bond. So the first peptide bond cannot be synthesized by any peptide. It has to be synthesized by nucleic acid. So there is an evolutionary link between the non-protein nature of the enzymes. But most of the enzymes we come across are protein in nature. Right? Are they heat stable? No. Being protein, they are heat labile. Mostly. They are heat labile. Beyond certain temperature, it loses its conformation and it loses its structure, function. When protein loses its structure, it loses its function. They activate any reaction by lowering the activation energy. Now, what is activation energy? Did you hear the sound? Now, what is it? What is the difference? What is the difference? Potential energy. The potential energy is converted into sound, right? Now, if the compound is here, substrate, and product is here, then it should come down like this. The problem is, this structure and these structures, which one will decide in which direction? Who will decide that A will be converted to B or B will be converted to A? Who will decide? Okay, sit down. Now, if I give you a bow and arrow, how do you use it? তীর ধনুক ব্যবহার করেছিস তো? বলো, করেছিস? কে করেছিস তীর ধনুক? বলো, নীলজামা কি করা হবে? কি করো বলো? হ্যাঁ, তুমি পোটেনশিয়াল টেনে ওটাকে করো, ওটা একটা স্ট্রেন হয়, তাই তো? হ্যাঁ, প্রথমে ওটা একটা স্ট্রেস হয়, স্ট্রেসের ফলে যে এনার্জি তৈরি হয় তাকে বলা হয়, যিনি ডিসিপেট করো তাকে বলা হয় স্ট্রেন। স্ট্রেন হলো তুমি স্ট্রেস দিলে ওর স্ট্রেন হলো, ও স্ট্রেনের জন্য একটা কি, যেমন তুমি পড়াশোনা করছো না, তাই তো? বাড়িতে বাবা খেলাচ্ছে, এখানে আমি, বাবা স্ট্রেস দিচ্ছে, আমি স্ট্রেস দিচ্ছি, তোমার কি হচ্ছে? স্ট্রেস না, স্ট্রেন, তাই তো? এর ফলে কি হচ্ছে? ধর পড়বো না, বই ফেলে দিয়ে বেরিয়ে গেলে, তাই তো? চলে গেল রামপুরহাট, তারাপীঠ, তাই না? তাহলে এই যে বেরিয়ে গেলে এর দুটো জিনিস হতে পারে। তো দ্য মোমেন্ট ইউ পুট দ্য টেনশন ইন দ্য bow before leaving the arrow, you have two options: leave it or bring it back, right? Huh? So at this moment you have equal possibility to release it or bring it back. That state is called transition state. A molecule cannot be converted to another molecule unless they reach a transition state. Correct? Now, here to here and here to here, there are two activations. Who decided this is product? According to some principle, all reactions are reversible, correct? So in some cases this will be substrate, in some cases this will be substrate, and vice versa, correct? So who will decide? Now, in our cell, it will be decided by how the brain will send a message through messenger. Who is a messenger? hormones. So depending on, suppose you are putting the bow, okay, putting it ready. Something is moving behind the bush. If it is a deer, you will throw it, but you find it was your dear one, not the DER. But what will you do? The moment you will see the face of the lady, then you will draw it back. Correct? So who will decide? Correct? What is the need? If we are hungry, some enzymes will convert AS to P. If we are satisfied, had a good lunch, then some enzymes will stop the conversion of AS to P. Correct? So this is called metabolic flux. Metabolic flux. It depends on metabolic flux in which side the reaction will go within the cell. Correct? And wherever it has to go, it has to bind with some enzymes, because in biological system, enzyme is necessary. Why? We cannot wait for millions of years to synthesize one molecule of ammonia. Right? One molecule. How many molecules of ammonia we produce in our liver? Innumerable number. Correct? You take more protein, production will be increased. You take less protein, production will be decreased. So efficient system we have. Correct? So what does it do? It changes a substrate to this transition state. Now this transition state is something which is neither equivalent to substrate nor equivalent to product. Correct? It is something new. Correct? Now, with time, this has to be increased like this in activation energy. How? Suppose there is a broth, you put on fire underneath it, start boiling. So you are providing energy from outside. Correct? Or there is a sail, there is a small toy boat with a sail. You blow it. So boat will start moving. You are putting some energy. So either you have to provide energy from outside or from inside. Now inside the body, who will provide energy? If you put a heater on my back, what will happen? Will it provide energy to my body? It will burn the face, only surface, the skin. It will never produce heat here. I am not metal. You know? Similarly, so it has to be inside the cell, and inside the cell, such extreme conditions are not possible. Severe amount of heat, severe amount of pressure, no. What is the pressure? Normal pressure? 1 atmospheric pressure. What is the normal temperature? 37 degrees Celsius temperature. Right? Within that, we have to work. And that's why proteins, what they do, they make a tunnel as if it is a mountain. You have to climb here. Now from here, you have equal possibility to roll down on either direction. Now, instead of going to that height, you might bring the ball here and bring it down. So what will happen? See, only this much energy is required. How it is possible? By putting strain on that molecule. By putting strain on that molecule. You bend the molecule like this. Bend the molecule like this. And then when you release, then that energy will be dissipated in the form of product. Right? This particular theory was called tunnel theory, as if someone is boring a tunnel through the mountain to reduce the activation energy. Effect of enzyme over effect of catalyst. Enzymes are something which is as if boring a tunnel through the mountain so that the substrate gathers less amount of activation energy and convert to product. Easy? Okay? These are all concepts which came little bit different from that of the chemical reactions in biological system. Unlike chemical reactions in biological system. Biological system এ আমাদের কিছু কিছু পরিবর্তন হলো, কি পরিবর্তন হলো? Moderate condition, less activation energy due to internal strain, molecule proximity, correct? And regulation. If you want to throw a ball, if you see the player, you will throw it faster. If you see not your player, you will not throw it. So catalyst versus enzymes, these are the points you must remember. Remember also this one, that this will never be asked in the examination. No one will ask all these things. But this is a basic idea you must have before entering into this particular chapter. So enzymes are organic and heat labile. They have high specificity, they have scope for regulation, they require milder conditions, and obviously they are much higher in consideration of speed compared to any chemical catalysis. Nature, mostly they are protein in nature except few which are called ribozymes. Now, what is this term apoenzyme, holoenzyme, this, that? You know? What is apoenzyme? What is holoenzyme? What is holoenzyme? If the enzyme has two components, protein component and non-protein component, the protein component is called, the non-protein component is called, if the enzyme has two components, then it's called non-protein component is called what is coenzyme? Huh? What is the difference between coenzyme and cofactor? Ke bolbe? Bol. Very good. Wow, brilliant. Ekdom correct. Thik. Coenzymes are organic in nature. They are dialysable. Correct? তাদেরকে বলা হচ্ছে coenzymes. Cofactors are loosely bound and cofactors are tightly bound, inorganic in nature mostly, and cannot be separated by dialysis. So the organic cofactors you have to remember like this. Most of the organic cofactors used in the enzyme are some or other types of derivative of water soluble vitamins. Water soluble vitamins mostly. What is this? Biotin, coenzyme. The dietary precursor is biotin, vitamin. Coenzyme A, pantothenic acid. This is 5'-deoxyadenosylcobalamin. Vitamin B12. Flavin, adenine, dinucleotide, FAD, FMN, they are all derivative of vitamin B2, TPP, thiamine pyrophosphate, derivative of vitamin B1, pyridoxal phosphate, PLP, pyridoxal phosphate, B6, correct? NADP, from niacin, tetrahydrofolic acid, from folate. Correct? All these you must remember, the coenzyme form and their precursor vitamin form. This is the organogram of structure of enzyme. Enzymes, simple, only protein, or complex, that is hollow enzyme. I said if it has non-protein part, then they are called apoprotein and cofactor. Apoenzyme and cofactor. Cofactor can be of two types: organic, inorganic. Organic, coenzyme. Inorganic, again there is a sub-classification. If they are very tightly bound, then they are called prosthetic group. If they are loosely bound but inorganic in nature, then normal cofactor. Correct? Say for example, zinc in carbonic anhydrase. It's a tightly bound. It's a part of the structure. That's why it is called metalloenzyme. Magnesium, whereas, is required for activation of hexokinase. Now, this magnesium is not a part of the structure of the enzyme. Hence, it is loosely bound. Correct? But not as loosely as coenzyme. So, it is just a prosthetic group. So, it is called metal activated enzyme. So, the metal part found in the enzymes, based on that, can be divided. Enzymes can be divided in two groups: metalloenzymes and metal activated enzymes. Metalloenzymes are those which contain metal ion as a part of their component, structural component. Correct? And metal activated enzymes are something which are required from outside for their activity. Now, how enzymes act? How enzymes act? There are three models. Lock and key model given by Emil Fischer, Koshland's induced fit model, right? And then this one, the latest one, preferential transition state binding theory. What is the difference? Now, as a matter of fact, this was discovered first, then this was second, and this was third, chronologically. Okay, but while reading, we will read this one first, then this one, then last this one. Why? Because there is a similarity, parity. Why? What is about parity? Lock and key model. Anyone can tell me what is lock and key model? What do you mean? What do you understand by this? Last bench, bolo. তুমি এই যে লাস্ট বেঞ্চে আগে বেঞ্চে কালো জামা কি বস বলো what do you mean by lock and key model? What is the material used for making lock and key? I give you three choice: wood, plastic, and metal. Why metal? What is there in metal which is not present in other two, wood and plastic? Rigidity. Correct? It will never change its shape. Neither the lock nor the key. Right? Lock and key, both are made up of metal. This also will never change, and this also will never change. For thousands of years, it will remain same. Suppose if you prepare this with a plastic, then what will happen? It will break when you put pressure. It will never fit again. Okay? So, Emil Fischer said that all substrate has got specific enzyme. No two substrate share same enzymes. Correct? Very good. Emil Fischer was a renowned chemist. No one could question whatever he said, like a dogma, you say. But problem arose when this question came. Suppose you are a believer of certain religion and certain forbidden meat in that religion is fed to you unknowingly. Can you digest that meat? Yes or no? Suppose unknowingly you eat the forbidden meat in your religion. Can you digest? Yes, you can digest very well. Right? The moment you come to know, then you cannot eat. But if you eat it, you can digest it. Correct? Then what happens when we change our religion? The God, the Almighty will change the genes in our body. The moment I will shift from Hindu to Muslim or Muslim to Hindu, our genes will change so that the moment I'll change to Hindu by mind, I cannot digest beef. The moment I will change to Muslim, I cannot digest pork. Because the capital He will change the genes. Correct? Because I am Muslim, I cannot digest pork. But the very moment I become Hindu, then in the next meal, I can digest pork. It is so fast. It is possible only in religion. Science cannot do it. Right? Can science do it? So what is the answer? Are they changing always, the genes? The genes are always there. So our enzymes do not differentiate whether it is beef or pork or something else. Fish or crab or snail, nothing. If the protein is same, having same bond, it will act on it, irrespective of our belief and religion. Doesn't matter. Correct? So these particular notion were directly against the first model, where neither key nor the lock can change their shape. Now they said that there may be one lock which fits with many keys. How? The key will bring about subtle changes, small amount of conformational changes, so that it will go inside and fit to different locks, or different key will fit to one lock. Which one? In this case, different key. The subset remain unaltered. The enzyme changes from one subset to another subset so that it can fit into many subsets. Why? Because the subset bond they are specific to is same. The bond, polypeptide bond present in beef, pork, meat, fish, egg are same. Nothing will happen, nothing will change. They can digest. Enzymes can work on it. Okay? So this slight changes in the enzyme, not substrate. Remember, substrate is not going to change. Enzyme will change its shape. So first one, neither substrate nor enzyme will change their shape. Lock and key model both are solid. Second one, the lock remains same. The key will bring about subtle changes so that it can fit to many locks. Third one is different completely. It was a very new idea. It was discovered in late 1990s, and it given a new concept. Okay. How many of you have seen the Oh My God movie? Akshay Kumar, Paresh Rawal. You have seen? So when Akshay Kumar first entered as and said like I'm God, what was the reaction of Paresh Rawal? Did he believe? Why? Because God was like you and me. He was looking like just you and me. He was not different. So what did he do? He appeared in the shape and size or in the color or in the attire seen in the calendar or photograph. Correct? So that's why. So God present in the photograph is the transition state. It has nothing to do in which form he is going to come and help us. Be it Almighty Allah, be it Almighty whoever it is. Suppose you are going to drown. Okay, one dolphin might come and save you. Savior. One fisherman can put a net and take you out of the water. In any form, he can come. Why he? There are seas also. Who knows what is the gender? It might be transgender also. Who has posed capital He? Why not capital C? And if why not capital C, then why not capital X? All are anyway. So that will be a different issue. So that capital He will come in any form, but that form is a transition state. And that form we hardly see them in our normal life. In our normal life, we see what? Either substrate or product. We cannot see transition state. Why? Because transition state is something which remains engulfed by the enzyme. When enzyme binds with the substrate, then only enzyme can see the transition state of the substrate. Why? Because enzyme also in native state bears a different structures. Say, this is the concept. Correct? Now substrate is seen by enzyme in some shape. Product is seen by enzyme in some other shape. Correct? This substrate is neither this one nor this one. It is different. So this enzyme is also different. So this particular theory is telling that both enzyme and substrate will change their conformation so that they can fit into each other in best possible way. Now, the example I was drawing, how do you conclude that example in this way regarding this? There is a bhajan like कौन कहता है कि भगवान आते नहीं तुम मीरा के जैसे बुलाते नहीं, है ना? बुलाने वाले चाहिए, खुदा जरूर आते हैं, सही तो? तमने की you must change, like Kabir, Sant Kabir, whoever it is, the lofty persons, whoever we had, Jesus Christ, you say, Hazrat Muhammad, you say, whoever it is, they change their lifestyle, they change their thought, they groom themselves in such a manner that they could see, they brought about subtle changes among themselves, that they see the God, the Almighty in that transition state. Like Paresh Rawal saw Akshay Kumar in the transition state. When did he see? When he read all the books, gathered the knowledge, it brought changes among himself, within himself. Then only he could see the God, not before that. Religious. So these are the three models. So what is the basic difference? Lock and key model, both enzyme and substrate are rigid, induced fit. Substrate rigid, enzyme keep on changing, small amount. Preferential binding, both enzyme and substrate change their shape in such a manner so that they bind in the best possible way. How did you find? What are the proofs? Now these are the diagrams should be accompanied with this theory when it comes to the examination. Lock and key theory, this kind of diagram you can draw and I can explain. Right? What next? Induced fit model. See, the active site is flexible, not rigid. Correct? So substrate remains same. Enzyme is changing. Now here another concept is very important concept is given in this diagram. What is that? Which one is bigger, enzyme or substrate? Enzyme. Is there any exception? Nucleic acid. Substrate is bigger than enzyme. Substrate is bigger than enzyme. Polypeptide. Substrate is bigger than enzyme. Polypeptide. Polysaccharide. Right? Now, okay, fine. But remember, the problem is the actual portion where the enzyme function, that is very small. Overall molecule might be bigger, but their active, where they function, the site is small. Correct? Preferential transition state binding, this is, you have to write only this particular sentence. Both enzyme and substrate bring about changes in their conformation so that they fit in best possible way with each other. Is called preferential transition state binding theory. This theory is proved by the following experiment. That is what the inhibitors designed, the inhibitors designed against the transition state of the substrate has more efficiency than in native structure. Correct? Now, naming of the enzyme. It was a jano, Sawai. Do you know about these classes? EC. This is what? Read out. Remember this. According to International Union of Biochemistry, all enzymes are classified into 6 or 7. Recently 7. The 7th one is added. Recently 20 years back it was added. The process completed last 5-6 years back. Process started 20 years back. It took 20 years to add one extra class. So the 6 classes are, what are the names? O, T, H, L, I, L. It is fixed. You cannot change it. EC stands for Enzyme Commission. EC stands for Enzyme Commission. Correct? So they will take few commission, a few money, and they will give the enzyme discovered by you a name. How? Like this. They will take few commission and they will give the enzyme discovered by you like this, 4 digits, starting with EC. EC, suppose there is a enzyme called EC1111. This is 2711 is given. Suppose this is 1111. Is there? Is there? Right? So what does it signify? This. The first one signifies a class. Which class it belongs to? OTHLIL, oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, and translocases. Seventh one, translocases. Now, T, second one, designate transferase. So it belongs to transfer, it transfers something. 7 is a subclass, the second digit. Transfer of what? Electron, hydrogen, what? Proton, phosphate, sulfate. So suppose this transfers phosphate. So in that scale is given. If it is phosphate, then it is 7. Right? So you put 7. Then subclass. From where phosphate is taken? Is it alcohol? Is it aldehyde? Is it acid? What? If it is alcohol, then it is acceptor. Who is taking? Where you are transferring? In some, who is giving electron? In other, who is taking electron? Acceptor or donor? In this case, who is accepting the phosphate group? If it is alcohol, then it is 1. If it is acid, it is 2. Like that. So in this reaction, it is like that. What is the name of the reaction you see? Then fourth one is specific name. What is the specific name? In every class, there is a sequence, chronological sequence of discovery of that particular enzyme. In that class, whichever enzyme will be first discovered will be named 1. The second discovered 2. Third, 4th. So this was the enzyme discovered in this group first. So given 1. It might have, suppose next is glucokinase. So rest all will remain same. This will change. 2712. 1111. What is the name of the enzyme? Alcohol dehydrogenase. Huh? So 2711 you know hexokinase, 1111 you know alcohol dehydrogenase. So all these are naming procedures. Okay. Very rarely they ask, but you should know. The first one is a major class, second one is subclass, third one is subclass, and fourth one is the chronology of discovery. Now this part sometimes they prefer to ask. So whenever you get this question, you have to first write down according to IUB, all enzymes are classified into 7 classes. Name them: class 1, class 2, class 3, class 4, class 5, class 6, class 7. Oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, and translocases. Then followed by this, in each one you have to explain. The first one comes oxidoreductases. So this is what? These are the enzymes which participate in reactions wherever there is oxidation or reduction. That means transfer of electron or proton. Any reaction wherever oxidation or reduction is taking place, this comes under this group. Oxidoreductases. Remember, always end this word in plural because there are many enzymes in this group. So group is always plural. One such example has been given here: lactate dehydrogenase. So lactate is being converted to pyruvate or vice versa, depending on the metabolic state. Which one is required in the body that time? If pyruvate is required, then this reaction will be preferred this side. If lactate is required, then this will be preferred. Correct? So in this reaction, energy will be utilized. In this reaction, energy will be trapped. So here, this one, you can say there are 2 types of examples. One is general example, another is specific example. The general example is like this. You can say A plus OH, it becomes AO, BH. like that, oxidation reaction. Transferases, enzymes which transfer certain functional groups. Right? So it might be amino group, carboxylic group, sulfur, phosphate, correct? But not electron and proton. Why? Because if you transfer electron and proton, then it becomes oxidation or reduction. For that, separate group is there. Functional groups should be proven, okay? Not electrons and protons. Transaminase, phosphate groups, all enzymes which transfer phosphate groups are termed as kinases, transmethylases or methyl transferases, transpeptidases or peptidyl transferases, transacylase means what? Which transfers acyl group, malonyl transacylase, acetyl transacylase. This is one example of alanine transaminase converting alanine to pyruvate and alpha ketoglutarate to glutamate. So one alpha amino acid to another alpha keto acid, corresponding alpha keto acid, and one alpha keto acid to corresponding alpha amino acid. Hydrolases. Hydrolase means splitting of a large molecule in presence of water. The enzymes which catalyze hydrolytic reactions. Splitting a large molecule in presence of water called hydrolases. Any digestive enzyme fall under this class. How many digestive enzymes you can name? Amylase, pepsin, lipase, face, digestive enzymes, bolo. Hydrolytic enzymes, কি কি আছে? Amylase. দু রকম অ্যামাইলেজ আছে, একটা হচ্ছে স্যালাইভারি অ্যামাইলেজ, একটা হচ্ছে প্যানক্রিয়াটিক অ্যামাইলেজ। বলতে হবে দে আর ডিফারেন্ট। স্যালাইভারি অ্যামাইলেজ অ্যান্ড প্যানক্রিয়াটিক অ্যামাইলেজ। আর কি আছে? আর কি আছে? ল্যাকটেজ, মাল্টেজ, আইসোমাল্টেজ আছে কি নেই? আসছি। এগুলি গেল কি? অ্যামাইলেটিক এনজাইম। এবার প্রোটিলেটিক এনজাইম। প্রোটিলেটিক এনজাইমের এক্সাম্পল শুরু করি। পেপসিন, ট্রিপসিন, কাইমোট্রিপসিন, কার্বক্সিপেপটিডেজ এ, কার্বক্সিপেপটিডেজ বি, দুটো আলাদা এনজাইম, আলাদা জিন। দেন অ্যামাইনোপেপটিডেজ, ট্রান্সপেপটিডেজ, ডাইপেপটিডেজ। দেন আরো আছে ইলাস্টেজ, কোলাজিনেজ, কারেক্ট? রেনিন হলো, আরো আছে। এবার লাইপেজ বলো। Let's see how many lipases you know. See how this is what is the name of chapter? Enzymes. So how many enzymes and names you know? If I ask you, this is the answer. If you know them, good. If you don't know, then you must write the answers. I am telling you the answer of the question. How many? Suppose you come to me or come to any external. He asks you, name how many enzymes you know. Only one question. Then come out like him. Trypsin, pepsin, amylase, lipase. Oh, he's like an extrapolar. What is the difference between you studying this much? Then lipases, name them. The major lipases are gastric lipase and pancreatic lipase. Correct? What else? We have phospholipases. Phospholipase A1, A2, B, C, D. Lipoprotein lipase. Correct? All these are hydrolytic enzymes required. nucleic acid, DNases and RNases. The multiple enzymes which can cleave RNA as well as DNA. Okay. So this is a complete list of enzymes in digestion. So when anybody asks you, name some enzymes, start naming like this. Then start with enzymes of glycolysis, then enzymes of pterocycle, then enzymes of tryptophan pathway, enzymes of phenylalanine pathway. How many enzymes are there? Thousands of enzymes are there. It's up to you how many you can remember. Correct? All digestive enzymes. Lyases. It is different. Why? It splits a large molecule in absence of water.